Hello everybody, I'm Alexy Khrabrov, the organizer of SF Spark and Friends, and here we are on location at Chartboost for a very special meetup on open genomics. It's called Scala and Spark for Good. Open genomics, mobilizing open source developers to work on cancer research together. And here with us we have Johnson John from Driver Genomics. He's one of the folks who actually introduced our community to the field. They are heavy users of Scala and Spark, and I let John introduce himself and talk a little bit more about it.
Thanks, Alexy. Yeah, so I work at a company called Driver Group. I was one of the co-founders. We are currently using Spark and Scala to start looking at mutations and in giant data cancer samples. So basically what we do is we collect samples from a tumor and a normal from a cancer patient, and we do a comparative analysis of those two samples, and with that we produce a free report that recommends basically which kinds of drugs those patients should go on. And what our company is doing is just giving these reports away for free in exchange for data. And we're building up this data and working on identifying drugs that will hopefully help these patients in ways that they're not currently getting help. So we're starting off with stage 4 lung cancer, and we plan on moving into other tumor types afterwards.
So what made you settle on Scala and Spark as your development stack?
Yeah, so a big part of it was the community. Just seeing a lot of interest in it and a lot of development. It seemed like there's a lot of building momentum. Another aspect was the type safety. So specifically in our report generation, it's really important that we don't make any mistakes when it comes to the different kinds of identifiers we have. We definitely don't want to mix up the case identifier with the patient identifier and accidentally give the wrong report to the wrong person. So having that kind of built-in type safety all the way down really gave us a lot more confidence in what we were producing. This
is interesting. It reminds me, so Kaiser health system, right? Before they dispense any drug, they need to scan your wristband. So this is almost implementing this in software, right? Like using the type system to keep track that you are not mixing up patients in their data, which is so this is kind of an interesting angle about type safety, which is I think it's a great application of type safety. But about the data size and speed, why don't you do it all in Perl, right? Like there are books, Perl for bioscientists and Python.
Yeah.
What technologically is different about Scala and Spark, which makes it easier for you guys to use them?
What I really like, so let's say we're going to do this all in Perl. It would be an extremely manual process. You'd be defining exactly where you split up the code, exactly how you parallelize, how you move the data back and forth. You'd basically be doing all that really low-level work yourself. And on top of that, Perl's kind of slow, to be honest. So the fact that Scala itself was a fairly fast compiled language, and it would do, with Spark, it would do a lot of this parallelization and distributive processing for you. It was incredibly attractive. So, yeah.
So Spark is basically an easy way to parallelize computation.
Exactly.
Great. So can you tell us a little bit about yourself and how did you basically come to, co-found Driver? Are you primarily a biologist, a computer scientist? How do you, see the fusion of these two tracks?
Well, I would say in academia, this fusion is starting to become pretty mainstream. So there are a lot of programs out there that merge the two. About 50% of the time you're doing biology, 50% of the time you're doing computer science. Actually, that's what I did with my undergrad. I majored in computer science and minored in biology. I discovered that solving these kind of biological problems using computers was just really interesting to me. I think we had this nice project where I was looking at HIV data, at mutations in HIV patients, and I was able to quickly find that certain mutations resulted in patients having lower CD4 T cell counts. And that was like just a really interesting thing that you could find just by looking at this data that wasn't necessarily obvious, and I'm not even sure if it was published at the time. But it was just a really fun kind of intro to what you could do with computers and biological data. So it went on from there, and I eventually got involved in a slightly different thing, different kind of biological data. I was looking at alligators and crocodiles and gharials and putting together their genomes for the first time. After you have a first genome, it's really easy to do a lot of different analyses, but putting together that first genome is really computationally intensive and very difficult. So I was doing that for these animals to help other scientific researchers with their downstream data analyses. And actually that is how I got involved in Driver. Yeah.
Interesting. So you mentioned this low counts. Can you explain to our audience, quantum biologists, what the low counts meant in that context?
Sorry, say that again.
You mentioned the low counts. And so one of your early problems you found.
Oh, CD4 T cell counts.
Yes.
Yes. So basically, CD4 T cells are a kind of immune cell that your body has, and one of the effects of HIV is that these immune cells are depleted. They're killed off by the virus. So what you can do is basically it's one of the things that makes you sick when you have AIDS. So you measure these levels, and what you want to do is try to keep these levels high.
Yes.
So basically by correlating the data, basically the data of the virus and what the virus's sequence is, with the outcome, how low the CD4 T cell counts were in a patient, you can basically, what I was hoping to find were these correlations, basically identifying mutations that would possibly result in more aggressive forms of the virus, right? So more aggressive kinds of HIV potentially.
Interesting.
Or maybe more resistant to drugs.
So this is an example of basically data science directly leading to discoveries.
Exactly.
Interesting.
Yeah.
Interesting.
And that was a toy project. It wasn't anything that turned into anything useful, but still, it was just really interesting seeing that kind of data and just exploring it myself.
So can you build on this? So there is this term personalized medicine, right? And you are in the frontier of this, right?
Yeah.
So what you are building is essentially an example. So can you explain, can you summarize for folks new to the area, what is the personalized medicine promise, and how you specifically going to achieve this in your space?
Right. So we're focusing on late-stage lung cancer right now, and I'll just go into this with a case. So one thing that came up a while ago was that there are about 7% of lung cancer patients have a mutation in a particular gene. This gene is called EGFR. So of those patients, you could just give them chemo and radiation. In that case, they'd have the same outcome as any other patient, any other lung cancer patient, which is not very good. By knowing that they have this particular mutation, and by knowing that that is the mutation that's driving their lung cancer, that particular mutation is causing their lung cancer to happen, there are actually drugs out there that match up with that particular mutation. So these drugs will actually target that gene and turn it off, so now the tumor can't grow anymore. So when we talk about targeted therapy, what we're talking about is a therapy that is very specific to an individual patient, right? This is the personalized aspect of this kind of therapy. So for those patients now that have this drug that's matched up with their specific kind of tumor, they do on average about 6 months. They have about a 6-month longer life expectancy than patients that just do standard chemo and radiation. And although that's actually quite a long time for these patients, 6 to 8 months extra for them to potentially, find another therapeutic option or, just finish up what they need to finish up in their life. I mean, it's huge for them, even though it's, yeah.
It's a start, right? I mean, given the time frames.
Yeah.
Usually, so basically that's a direct result of doing proper match of individual case and already available medicine. So this is what exists today.
Yes.
Right. But going to the future, can we kind of see a world where we have much more drugs and we kind of match them individually? What would it take, to get that? Like, do we need is it just a computational problem, or is it also a governance problem? Like, how the pharma companies fit in there, and what is your plan to connect all of this?
Yeah. I think there's a really big collaboration problem right now where, every doctor and every lab wants to have their own data and wants to be the hero and discover something. So what you have are a lot of different siloed data sets that are very specific to different centers. And these siloed data sets, they don't get shared. You don't have a lot of people collaborating on discoveries. There are a couple public efforts that are working on getting past this, but they're pretty minimal. They're not, you don't get hundreds of thousands of patient samples. You get maybe 1,000 or 2,000 in a particular tumor type, and those are the bigger projects that are out there. So I think that is partially holding the field back. So I think that something that opened up the data, and also open, collaborative, potentially even competitive analysis consortiums, like different kinds of Kaggle-style competitions. I think those kind of things would be huge, right? Because the alternative is you publish your own paper and you run your own competition. And there is so much room for either, direct or indirect dishonesty, or just not trying as hard to make the other people's methods work, and it doesn't really push the field forward.
Not to mention duplication of work, right? So this actually very nicely kind of segues into our meetup topic. So we had this informal group started at Text by the Bay, where we have folks from 2 universities and 3 startups, right? I think most of them are here today. And we have a wider group of developers, and the response which we get, presenting them with this is always very enthusiastic. So basically, it's a lot of people say, how can I help? A lot of companies say, how can we help? For instance, Nitro, my company, has a program called Nitro Gives, where we give developers and employees in general 5 days a year to volunteer on projects of their choice. And also, as a company, we can put resources behind this, right? And I think a lot of companies in the area and beyond have this kind of programs, right? So potentially there is a lot of resources on individual time. In your spare time, you can choose to build a pet store on Ruby on Rails, or you can help with Adam in Scala and Spark, right? And we hope that we can enable you, but Adam and Spark is not pet store, right? Like everybody understands, how you can buy cats and dogs. Not everybody understands how you identify mutations. So what do we need to do collectively? How can we go about providing these developers with resources? And let's say you have 1,000 developers at your disposal, which I think you will, given the trajectory. Basically, but for now you can have them through spare time, and kind of you will have partial time of 1,000 developers at nights and weekends, and in open source setup. What's the most effective way to utilize these 1,000 developers? How would you structure their work? How would you structure the community hub so we can kind of separate tasks, publish interesting things? What are the good starting points for this community resource?
Okay, so I think what would be amazing is if we could get everyone doing Kaggle-style competitions, releasing their code, and putting their code onto platforms that are really easy to integrate into large, enterprise-style environments. So if they could do their code in Scala and Spark the first time, that would be excellent. Even if they just implement their algorithms in other languages, and then other developers can come and re-implement them in Scala or Spark, that would also be excellent. But I think getting more people participating in the competitions that exist, and also generating new competitions, which I think is going to be there's going to be a lot of, you're going to have to get a lot of government approval for this. You're going to have to get patients that agree to release their data in open ways. There's a lot of work to be done, but getting more people trying out their ideas on these data sets and trying to make discoveries and trying to just getting the plumbing to work, and like you said, contributing to Atom and Avocado and these other tools, just getting that data movement stuff before even the data science aspect of it. I think those are all important.
And this is the area which a lot of data engineers can know very well, right? So I think that can be an entry point for a lot of guys here, how we set up the data pipelines, right? So this is great. So we're looking forward to your talk, and we're looking forward for the developers to collaborate with Frank and John and get this field moving forward. We have the new community resource called opengenomics.io. We'll publish all the links there. So if you want to participate, join our mailing list, follow the discussions, and hopefully we'll get this whole community going.